In this video, we will discuss about this GlyTR2, that is density dependent specificity of GlyTR2 towards GlcNAc containing TACA, that is tumor associated carbohydrate antigen. In the previous video, we have discussed about GlyTR1, whose specificity is more focused on beta 16 GlcNAc branched and Glycan. However, they also optimized another another car that they called as GlyTR2 that has a specificity towards TACA that contains GlcNAc or 5-N-acetylgalactoasmine containing TACA, that is tumor associated carbohydrate antigen. And this is what their optimized structures looks like. That has, in this case, they use stiff linker, 4 stiff linker, and they also use the CD3 antigen single chain variable fragment and this his-tag and this CD301 which is attached via stiff linker, and then they attach this and then they transduce this CAR into the T cells, and this CAR T cells can then act on the cancer cells that contains this tumor associated carbohydrate with high density of GlcNAc or 5 GlcNAc containing tumor associated carbohydrates. And thereafter, once they form their CAR Ts, they check its CAR Ts whether it exists in multimer, monomer. They are able to see that most of these CAR Ts are exist in the monomer, and they have validated using the size exclusion chromatography and also using the silver staining and Western blot. Thereafter, they checked it with by co-culturing these T cells with the co-culturing this. First, they checked this GlyTR2 binding with different antigens, that is TN antigen, GalNAc antigen, GlcNAc antigens, and what they are able to see that when they check the TN, when nothing is present, it shows this 100% binding, and when these TN antigens are present, this binding is reduced. And when this GalNAc is present, in this case they co-culture it with the CD3 minus Jurkat cells. So CD3 does not have specificity. They check the specificity of this lectin binding using the GlyTR2. So when this nothing is present, then it shows a GlyTR2 binding like around 100%, and when these TN antigens are present, the GlyTR2 binding reduces. Nothing is present, it means that now no competitive antigens are present in the sample. So these CAR T cells can completely work towards these cancer cells. And when these GalNAc antigens are present, when the TN antigen presents, this binding reduced because some of the TN antigens are being utilized. TN antigen sites on the CAR T cells are being utilized by this TN antigen. And when they use GalNAc, the sites are also reduced, but when they use GlcNAc, there is no reductions in the sites they have observed. And they also check it with this Gale negative Gale knockout cells, and these they are able to see that in the case of Gale knockout, they are not able to see any binding. And once they validated their results, they now co-culture it with these cancer cells, that is breast cancer cells, and they are able to see that increase in the cell death in the 3 donors samples that they have tested. And in this case, they vary the amount of GlyTR2, and in the panel G, what they did is they used effector to target ratios around 10:1, and they are able to see that when the high antigens are present, the binding is high. When the low antigens are present, in the case of clone 12 and 11, the binding is lower, indicating that with high density, the binding efficiencies of the GlyTR2 is high. And when I check it for the cell death, in that case, they see the relative increase in the cell death where the binding efficiencies are high, and lower cell death where the binding efficiencies are lower. Thereafter, they check the activations of the T cells on the CD8 plus T cells. They are able to see that when the donors are present alone, not in co-culture with the cancer cells, they do not see high positive cells. In this case, they use the effector target ratio around 5:1. And when they use the cancer cells, when they go culture these PBMCs with cancer cells, they are able to see high activations of the T cells that can be seen in this panel eye. So this uh, results further indicates that the second optimized GlyTR2 CAR, which has a specificity towards the TACA that contains 5 GlcNAc or N-acetylgalactoasmine carbohydrate structures, seems to be working well, and it has high efficiencies when they check it on the patient samples. And we can see that its activations is the cells are more activated when they are present with the cancer cells. However, when they are not present with the cancer cells, the activations are low. So this will have less side effects on the healthy cells also. So this is what they have presented in the main figure 2. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.